good evening, everyone. Myself, Jay. Today, I would like to introduce our today's speaker, Dr. Sarita Mahajani. And for those people who are new here, I would like to tell you this is ongoing webinar series, and today is our third and last session. So, let me introduce the speaker quickly in interest of time. So today is our speaker, Dr. Sarita Mahajani. Dr. Mahajani had done her BSc Microbiology from University of Pune. Ma'am has done her MSc Microbiology in 1980 from Shivaji University, a PhD in Biology from Dr. Baba Saheb Ambedkar University. Ma'am's current position is Principal Consultant and Scientist at Chitale Dairy, Sangli, Maharashtra, and Ingersoll Local Dairy, Ontario, Canada. Ma'am has spent 24 years in University of Mumbai as a lecturer and as a professor, and out of which 8 years she has spent as head of the Department of Biotechnology. Ma'am has served 5 years in University of Pune as HOD Biotechnology, and other than that, Ma'am has also served as a principal in Biotech College, Maharashtra. Ma'am has also been associated with Singer College of Engineering and SSP University of Pune uh, as a assistant professor, as a adjunct professor, and also as a visiting faculty member. Ma'am has also served as a consultant in Amphenol Interconnect, Pune for 4 years, and Ma'am has published so far so many research papers in international peer-reviewed journals, and Ma'am has also been poster presentation judge in many international competitions. Ma'am is currently on board peer reviewer of respected Elsevier publications, and Ma'am is also currently advisory board member of International Journal of Biotech Applications, and Ma'am is associated with various international and national organizations around microbiology and biotechnology. And so, in short, I would like to conclude that we have someone with more than 4 decades of rich experience. I would like to welcome Dr. Sarita Mahajani.
Thank you, Jay. It's nice to see you back again. I'll share my slides now. Yeah.
Sure. Thank you.
Can you all see? Okay. Hello and good evening to one and all. Hope you all are keeping safe. Hope Professor Shilpa is recovering. Pray for her speedy recovery. Thank you so much for inviting me for this series. The series started with recent big trends in biotechnology. We started with the vaccine candidates for the first lecture and reverse technology used in vaccinology for the second one. And today we are going to touch upon the other interesting tools and techniques used in industries. The life sciences have made great advances in the past years, and biology, life science, and the mega trend of digitization, computerization are growing closer together, enabling enabling the innovations that impact our daily lives in greater scope. We call it as biorevolution. So intuition begets in innovation, and in the post-pandemic landscape, we can see that technological companies are positioned to make substantial impact on the markets and the societies. Innovation is surging ahead, and now more than ever, technology is primary tool or primary force shaping how we grow and live. The technological advancement and innovation have been occurring across industries, a trend that has been true in biotechnology also. In light of COVID-19, this has been even more true because biotechnology is the core of understanding the virus and developing treatments and solutions. A range of scientific research streams are coming up, and they are nowadays collectively called as omics, OMICX. So what does omics means? It's a collective term for technologies that allow the comprehension or comprehensive identification and quantification of the complete set of molecules. For instance, we can say proteins, carbohydrates, and lipids, of the biological system, that is cell, tissue, organ, biology.
Hello, excuse me, ma'am. I think you are not audible just now, and also your screen is not visible.
Yes, yes, 200, yes. Madam has to unmute yourself.
Yes. Ma'am, you have to unmute yourself. Somehow you are on mute position.
Oh, can you see now? Can you hear me now?
Yes, yes, we
Yeah,
can.
ma'am. Yeah.
And can you see the screen?
Not actually.
No, ma'am.
Okay, I'll share it. Can you see?
Sorry, no luck so far. Okay, we got it now.
Yeah, yes.
Okay, is it okay?
Yes, yes, it is very much visible.
Yeah,
Okay,
I
can
am.
you hear me?
No,
Can
I
you
think
hear me?
slides are not visible.
Yes, yes.
I've already shared my
Yeah,
screen.
now visible. Okay, okay, fine.
Okay, so we were saying that omics is a collection term, like, set of technologies that allow the comprehensive identification and quantification of complete set of molecules. For example, proteins, carbohydrates, and lipids of a biological system. If you say biological system, cell, tissue, or organs, and biological fluid, or even microorganisms at a specific point of time. So omics and molecular technologies is defined to cover the study of omics as well as technologies to engineer, design, synthesize, modify the same, that is omics. So coming to the first slide, which is discovering the biorevolution. So it is the biorevolution that is defining innovation in the life sciences. How this might be a game changer, all the techniques that have come out, all the tools that are being used, and all this has led to new ways to understand and explore biology. The range of life sciences on Earth is incredibly complex and diverse. However, the methods to analyze them can be remarkably similar. Technologies and methods are transcending uh, disciplinary boundaries even faster. As a report says here, it gives you in tabular form very nicely. The wave of innovation is being enabled by advances in biological sciences, accelerated by developments in computing, data analytics, machine learning, then artificial intelligence, and biological engineering. So all the areas are being integrated or coming together now. And the four arenas that they have marked here in this report is the which are biorapidly advancing are biomolecules, biosystems, biomachine interfaces, and biocomputing. So if you define the definition of biomolecules in this report covers the mapping and measuring of intracellular components, for example, DNA, RNA, and proteins in the study of omics. This also includes the engineering of intracellular components, for example, the genome editing, the gene therapy, or monogenic diseases. The definition of biosystems, if you say, it covers engineering at the cell, tissue, and or the organ level, including stem cell technologies, transplantation that are used, and for example, the cultured meat grown in the lab. For biomachine interfaces is a field of biology defined as the connection of nervous system of the living organisms to machines, including in brain-machine interfaces. For example, hybrid systems that are used connecting nervous system of the living organisms to the machines. And the last one, biocomputing, it's a field of biology defined as using cells and cellular components for computational processes. For example, storing, retrieving, processing data, and data storage in the strands of DNA, that is the example. And if you go further, you can see each and every technique or tool that is used by the industries. And these are the emerging ones which are coming up now and mostly used to have innovation. We come to the oldest one by inventing recombinant DNA technology. The famous Boyer and Cohen jump-started the biotechnology industry in 1973. So rDNA technology was invented largely through the work of Boyer or Cohen and Paul Berg. The technique was to isolate pieces of DNA from an organism's genome, splice them with other pieces of DNA, and insert the hybrid DNA genetic material into another organism, such as bacterium. Pieces of DNA can be engineered in a fashion that allows them to be copied, replicated in bacteria or yeast. This involves attaching appropriate elements to a piece of DNA and then transferring it into bacterial or yeast cell. With those elements, which then instructs the bacteria or the yeast cell to copy this DNA along with their own. So the process is also known as DNA cloning, with the resulting cloned DNA referred to as recombinant DNA. So recombinant DNA sequences can be placed into vehicles called vectors that ferry the DNA into the suitable host cell, where it can be copied or expressed. So in 1978, Boyer and Itkura also constructed a plasmid that coded for human insulin and produced synthetic insulin. In August 1978, they published their report, and by using new transgenic genetically modified bacteria. In the case of recombinant insulin, there were companies which came together. It was a joint venture agreement between Eli Lilly, Genentech, and other companies to develop the production process for this particular synthetic inulin, which they called it as Humulin. So in 1982, Humulin was approved by FDA, and it became the first biotechnology product to appear on the market. A few interesting or notable applications in the areas are given in the table, which you can see on the left-hand side. And this genetic engineering, which is exploiting the huge potential of microorganism, plants, animals for the restoration of environment, that comes out first. Then actively involved in the development of microorganisms and catalyst for a remediation of contaminated environments. In the development of eco-friendly processes, such as developing recombinant strain for the biofuel production, etc. So the production of extra nutrients in the food, increased growth rate, disease resistance, then herbicide resistance, better taste, increased shelf life, etc. These are few of the things that recombinant technology takes care of. Then lesser requirement for water, then genetic engineering applied to plants and animals, to human production of antibiotics and vitamins and insulin, etc. So as per the forecast 2020 to 2025, the recombinant DNA technology market is growing continually, mainly due to the commercial success of the technology in biopharmaceutical industry. So this is the, if you say as emerging, we can say that this is the oldest technology which started quite long back. Then coming to the next one, and a most fantasizing one, or you can say it's a most interesting technology, exciting everyone, and read about everywhere, and there are lots of papers coming up, and so many industries are using this particular tool called as CRISPR or Cas9 system. So CRISPR is clustered, regularly interspaced, short palindromic repeats. So we have CRISPR, which refers to sequences in the bacterial genome. They are repeated sequences with a spacer sequence in between. They afford protection against invading viruses when combined with a series of CRISPR-associated proteins, which we call it as Cas proteins, CAS. So Cas9 was the first one which was built, and it is one of the associated proteins. It's an endonuclease. You know, endonuclease, it cleaves, it cuts both strands of DNA. That is the function of nucleases enzyme as a totally, if you say. Cas9 is directed to its targets by a section of RNA that acts as a guide. This tool uses a small piece of RNA, you can say, which is a short guide sequence that attaches to a target sequence of DNA and to a Cas9 enzyme. So the enzyme nuclease is being guided by a RNA, short RNA. And this RNA, you can structure it, you can construct it, you can tailor it. So Cas9 enzyme cuts the targeted DNA at the targeted location, which enables genetic material to be added or deleted. So this RNA-guided endonucleases CRISPR-associated protein Cas9 in particularly has attracted attention for its promise in basic research and gene editing based therapies, because it can accurately cut and alter the DNA with targeted specificity and relatively easy implementation. In the first 7 years, CRISPR gene editing potential was unleashed on the world. Scientists have used to make jointless tomatoes, fungus-resistant bananas, and higher yield of corn, soya, and wheat. And they used to develop apples resistant to fire blight. And the reports on in about 2019, one report in September 25th, it was published. This gene editing technology has been promoted widely by scientists in the promise of food and agriculture. It is used to edit the genes of crops to make it more nutritious and tasty. This is one of the best innovations, you can say, made in retail industry. Next, we have Casper, which takes on blood disease. And then we have an interesting and very new report which is coming up with a clinical trial that had taken place 1 year before. A patient with sickle cell anemia, Victoria Gray. Sickle cell disease is caused by a genetic mutation, as you all know, that produces a defective form of hemoglobin, the hemoglobin that carries oxygen to our blood. And the defective hemoglobin turns the red blood cells into deformed, or you can say sickle shaped or crescent shaped. That is why it is called as sickle cell anemia. So this particular cells, they grow in different shapes, like, and these defective cells don't carry oxygen well. Then they become hard, they become sticky, and they tend to clog the bloodstream, jamming it or clogging it. And because of this, there is attacks of pain, damaging vital organs and different parts of the body, and often premature death. So for experimental treatment, in this particular patient, if you can say it was a clinical trial, scientists removed cells from the patient's bone marrow and used a CRISPR technique to edit a gene which enabled the cells to produce a protein known as fetal hemoglobin. Now, fetal hemoglobin is made by all fetuses in the womb of the mother to get oxygen from the mother's blood, and it usually stops being produced by after birth, about 2 to 4 months of the child. So this was used, and the hope was that restoring production of fetal hemoglobin would compensate for the defective hemoglobin produced by sickle cell patients. So Victoria Gray received this treatment on July 2, 2019. Billions of her own stem cells were removed. They were reprogrammed with CRISPR to produce healthy blood cells and then infused back into her. So this promising change needed to produce fetal hemoglobin, indicating that the edited cells were continuing to grow, survive, and function in the body for a sustained period. This was the first person with the genetic disorder to get treatment in United States with the revolutionary gene editing technique called CRISPR. And as 1 year anniversary of her landmark treatment approaches, she received the good news that billions of genetically modified cells the doctors infused into her body clearly appear to bring down the complications of the disorder and to keep her out of all the pain and all the disorder complications. So this was a great history. You can say it was a great innovation. So this is the same one, CRISPR one. So 2020 was breakout year for the CRISPR disease detectors, which hit the market. The application of CRISPR technology as a diagnostic tool came up in the market during this particular period. The pandemic sped up the, you can say, the need for the test. The Sherlock CRISPR SARS-CoV-2 kit is the diagnostic first kit that was found out, and for the infectious diseases caused due to COVID-19. In May 2020, FDA announced the emergency use authorization for the Sherlock Biosciences Inc., which is a CRISPR-based. Sherlock stands for Specific High Sensitivity Enzymatic Reporter Unlocking. So it again uses the same technique, that is CRISPR technique, but this is a very easy tool to be used. The assay, as you can see in the diagram here, is comprised of two steps. The nucleic acids are extracted from the patient's specimens. The step one is reverse transcription, making cDNA from the complementary DNA from the RNA before running the reaction. The reverse transcription loop-mediated isothermal amplification is a one-step nucleic acid amplification method to multiply specific sequences of RNA. So they are using reverse transcription, which is isothermal, that is at one particular temperature. Unlike PCR, which uses different temperatures and different steps, this is only one step. So this was the amplification step that was used. It does not require thermal cycles. As I said, in just like PCR, it doesn't require. And the step two was the transcription of amplified DNA to activate collateral cleavage. Now here the cutting takes place by the CRISPR complex program to the targeted RNA sequence, exposing them, and then when the CRISPR cuts and exposes the RNA targets of the virus, and the targets were open reading frame 1AB, or and the nucleocapsid N, where the viral targets that were used that are used to diagnose this particular virus. And these, in the process 2, you will find that in the step 2, you'll find that is detection of the viral RNA sequence using Cas19. So during this particular step, there is also one thing that is done here is the cleavage of nucleic acid reporters results in a fluorescent readout detectable by a standard plate reader. So it fluoresces if they are the same COVID targets. A dedicated instrument platform, that is thermal cycle, is not required here. And that is why this is much easy. It's only 2-step. Now they have a different, another one which is coming up now, which is called as stop Sherlock testing in one pot, that is one shot, and it is called as stop COVID. And it also uses CRISPR program, and it uses Cas13, and it is one step. So they are still coming out, and they have to receive the authorization. And coming to the next one, last but certainly not the least, in October 2020, Nobel Prize in Chemistry was awarded to 2 women scientists, Emmanuel Carpentier and Jennifer Doudna. So they received this particular award for this technique which they had devised, that is genome editing. Since its arrival in 2012, scientists have published about more than 300,000 studies using the tool to manipulate the genome of organisms across every kingdom. So including mosquitoes, tomatoes, King Charles Spaniels, which is a dog in which the dog had the muscular atrophy and it was cured. So gene editing was done in an even human. So it's cheaper, faster, easier enough for almost everyone to use. The win of this particular award also broke barriers of another sort. The Doudna and Carpentier are the first women to win a Nobel Prize in the sciences together. Many women think that no matter what they do, their work will never be recognized the way it would be if they were men. So Jennifer says that upon learning the news, I think it refutes that. It makes a strong statement that women can do science, women can do chemistry, and that great science is recognized and honored. In other words, she continues saying that women rock. We couldn't agree more. Yeah. So coming to the another technique which is used on great scale is next generation sequencing. It has proven to be an invaluable tool for investigating diverse environmental and host-associated microbial communities. In this particular technique, mostly you see that there is interactions that are read or understood, helping to generate enormous new data sets that can be mined for information on the composition and functional properties of vastly great numbers of microbial communities. So microbial genomics is largely the identification and characterization of their genetic compositions. The ability to process, analyze the genetic or genome data collected and microbial organisms in the corners is the cornerstone of modern bioinformatics. So its broad applications cover every sector of our lives, such as ensuring safety of the food supply, maintaining human health and wellness, then countering the spread of disease and protection of the environment. With bioinformatics tools developed and in place, all aspects of microbial genomics can be analyzed. We can identify microbes, assess microbial populations in the environmental places, catalog environment evolutionary pathways, and define genetic relating them together between the microbial strains. Furthermore, research is underway to explore the potential of using genomic trials to ascertain antibiotic resistance and even virulence of a microorganism. If you read the diagram here or the figure here, you will find that how they are related to each other and how they bring about the interactions in the community. For example, if you say different sequencing and bioinformatics used for human microbe analysis has been shown here, one example which I got from a report. So in the 16S recombinant gene ed profiling, what we do is we take the raw sequences, obtain the and pass it through quality filters to minimize the presence of sequencing artifacts. So the resulting filtered sequences that are reads that we get are clustered together then into an operational taxonomic units, which you call it as OTU, which represent similar organisms. After this taxonomic identity or identification is assigned based on sequencing homology against the 16S RNA gene databases, and relative abundance of these particular OTU or operational units is calculated for each sample. So the resulting, what you get here is the table is also used to quantifying populations, diversity within and between the samples. So you can see the relationship between each sample here or each microbial community here. And the techniques that are used, you can see later on related to the 16S RNA. You often bring that also together. So you have shotgun sequencing is a laboratory technique for determining fragments that are sequenced individually. The method involves breaking the genome into collection of small DNA fragments that are sequenced individually. Metagenomics is a study of genetic material recovered directly from the environmental studies. And metatranscriptomic, the science that studies gene expression of the microbes within natural environments. So all these are, when you do all this, you bring in the interactions together. Thus the relative abundance of each active pathway or gene or microorganisms in the human microbe can be determined. The applications of next generation sequencing in microbial community profiling can help to answer questions like, who is present in the community, what could they be doing there, and what pathway they are using, and how these microorganisms are interacting with each other. So you see all this relatedness when you apply all these techniques together, which you call it as next generation sequencing or the genomics. Then there is a famous or very interesting technology that is used, or a tool you can see that is used. It is synthetic biology. Synthetic biology is nothing else but a logical extension of what has been called as recombinant DNA technology or genetic engineering since 1970s. The oldest application of biotechnology is biomanufacturing of insulin. Scientists discovered how to co-opt yeast and bacteria into producing human medicine decades ago. But while producing insulin, it requires only a single gene. There are many important medicines such as Taxol, which is an anti-cancer drug, that requires entire pathway to be studied. And that's where synthetic biology comes in. It is the field of science that involves redesigning organisms for useful purposes by engineering them to their new abilities. It uses molecular methods, of course, to construct DNA-based devices that perform new functions. It is divided into two areas, you can say, the entire genome engineering, especially on how to produce and manipulate whole genome sized. And then the second one is how to get them into the cell. It uses 4 principles not typically found in genetics, genomics, or molecular biology, that is abstraction, modularity, standardization, designing, and modeling. So it is more computer based. And in some ways, synthetic biology is similar to another approaches like genome editing, because both involve changing the organism's genetic code based on how the change is made. In synthetic biology, scientists typically stitch together long stretches of DNA and insert them into an organism's genome. These synthesized pieces of DNA could be genes that are found in other microbes, or could be entirely novel also. Genome editing tool can also be used to delete or add small stretches of DNA in the genome, so that so researchers synthesize an organisms and so then there's a question that can they synthesize the entire genome? The answer to this is of course yes, and it has already been done. In 2002, scientists in United States synthesized a viral genome for the first time. The viral genomes are much more smaller as compared to bacteria and other microorganisms, and they showed that it was possible to create the polio virus from scratch, and brought attention to the risk that synthetic biology could be used to develop biological weapons. While this group of researchers did not intend to cause harm with their research, their work understandably raised concern that bad actors might use synthetic biology to malicious purposes. So the article in 2017, which you can see there, I've mostly put all the articles there, and the references are given at the end of the slide. So synthetic biology is revolutionizing the biotech industry, and it is increasingly applied to previously thought of markets. Here the discussion is done on importance of the industry to the bioeconomic, and there are two keys: synthetic biology approach to research and development, and the unique nature, which is carefully designed, that is stakeholders, inclusive, community-derived evolution. So you can find this in this paper. And there is also one paper in very recently published in 2020, which is of Biochem Society. So this is developing synthetic biology for industrial biotechnological applications. So the startup investment, the segment technological approach, the engineering biological systems that are necessary in the market now, and how they are coming up, everything has been given with good, with all the startup companies that you find going on now. Coming to the another important and another interesting, I did not say much about it because in the last two lectures we have already dealt with it, that is nanoparticle or nanotechnology. How about a mobile phone fitted with a lab on a chip that can diagnose illness? So nanotechnology is already serving lives, and it will be crucial henceforth also. It is defined as the manipulation of materials that range from the nanometer to the micrometer scale. For a comparison, the cells of living organisms are typically around 10 micrometer across. So that puts the machinery and the components of the living cells within the nanoscale size range, making them ideal for interaction with functional nanoparticles and nanomachines. And bio-nanotechnology, also called as nanobiology, also called as nanobiotechnology, are the same names. That is the convergence of nanotechnology with biology. So the major aim of bio-nanotechnology is to create tools that may study living molecules and at a smaller scale. So it's union of nanotech and biology. So the March 2020 report, which is given an introduction of bio-nanotechnology. So this particular report gives you a brief on all the nanostructures that are used, and they have compared the two ones, that is the chemical methods that are used formerly, that is ion sputtering or solvothermal synthesis, or the sol gel technique, which has two basic approaches, that is bottom up and top down. So these traditional methods, they say they release hazardous substances to the environment, so they are potential to human exposure with the nanoparticles will also increase. And they have given here that they have attempted to reduce this particular hazards and the risks, you can say, by using new eco-friendly agents, for example, microbial enzymes or plant phytochemicals instead of chemical agents. So a non-toxic way of synthesizing nanoparticles can be achieved by using a green method, which is also called a biological method, which is an inexpensive way for manufacturing and synthesis. There is another paper report which is of just recent April 2021, which is still going to be published now, and it is in Food Chemistry journal, Advances in Applications of Nanotechnology in Global Food Industry. So here they elaborate on various types of nanostructures used in food industry, bringing about changes in the production, processing, packaging, transportation, consumption of in the quality of food. So its application in food processing and food storage is numerous. People all over the world are using it, and there was another paper in Nature Biotechnology, which was an older one, which had already predicted this, April 2010, that it was a groundbreaking paper by Lenhart, a lead author, and he, at the age of 32, he developed, and you can see the diagram in the center, which is a dip pen nanolithography. So he developed an internationally recognized innovative work in bio nanotechnology, related process, you can say, dip pen nanolithography, DPN it is called, which uses a sharp pen-like device and ink to write nanoscale platform patterns on solid surfaces, capable of producing materials with enormous potential, not only for diagnostic applications in healthcare, but also tissue engineering, drug discovery, or you can say computer chip fabrication. So in other words, it is a big deal technology on a nanoscale. It encompasses or compromises objects that measure just 100 nanometers or less in at least one of the dimensions. So the special point to be made here is about the step which we are going to see into details how the industries are using in the next few slides. So coming to, after going through the emergent technologies, a few of them which are very recent and often used, now I would like to go touch upon few industries which are using these techniques. And one of it which I like profusely is this one, San Francisco based company, which is called as Perfect Day, which was founded just 5 years ago by two vegan scientists, Ryan Pandey and Perumal Gandhi. So their goal was to develop a kinder way, a greener way to make animal-free dairy products people already love. So the co-founders or the founders, Ryan and Perumal, enjoy a taste of their world's first animal-free ice cream. You can see the picture on the left-hand side. And it was in early 2014, the two of them, recently struggling vegans, and the history goes long back. They have something very much in common. Ryan studied chemical and biological engineering at Tufts University, where he worked on tissue engineered meat for his graduation project. And Perumal studied biomedical engineering at SUNY Stony Brook, where he worked on a project to develop functional tissue engineering scaffolds for medical industry. And prior to that, Perumal has studied biotechnology at Satyabhama Institute of Science and Technology, Chennai, India, and worked as a research assistant for the Srivastava lab at IIT Mumbai. So these two founders have a lot in common, as I said, both of the same age, both bioengineers with an Indian heritage, and both of them frustrated new vegan and aspirations for making animal-free milk. The duo put their combined engineering backgrounds to work, embarking on a journey to discover how to create dairy using well-known fermentation techniques. Together, they set out to create a company to revolutionize the dairy industry, and thus came the name called as Perfect Day Company or Foods, Perfect Day Foods. Embracing the serendipity, they immediately agreed to team up and they build the protocol together. So their main issue was cows are great at making protein, both for as meat as well as milk. If all you want is a cow protein, why grow the cow? They came up with the way of using age-old process of fermentation to convert sugar into whey and casein, the two main ingredients in cow's milk. how microflora can be used to produce animal-free ingredients of milk, dairy, cheese, and ice cream. This is what they show, their company shows. They recently whipped that whey together with fat, sugar, and other ingredients to create 1,000 pins of milky chocolates, vanilla salted fudge, vanilla blackberry toffee ice cream, etc., etc. They have lots of products in the market now. They were named as Forbes 30 under 30 in 2019, committed to make delicious, nutritious food that does less damage to our planet. Luckily, they stumbled upon a study by the two. Now, after they got a little bit of seed funding, a little bit of protocol was put up, and they came upon what the name of the company should be. Formerly, they started, if you read their history, they started with Bufri name, but later on they stumbled upon a study. Two dairy scientists who discovered the calm, happy cows produce most milk. The cows are happier when they listen to music. That is what the scientists said. One of the song was "Perfect Day" by Bio Reed, and it's a beautiful song if you hear it, and it seemed like a perfect fit here. The song, if you want few lines of it, it is drink sangria in the park, and then later when it gets dark, you go home. Just a perfect day. Feed the animals in the zoo, later see a movie, and then go home. Oh, it's such a perfect day. And the last line say that you made me forget myself. I thought I was someone else, someone good. Oh, it's such a perfect day. I am glad I spent it with you. Such a perfect day. We are going to reap just what you sow. You are going to reap just what you sow. So it's a beautiful tune song, and they took this theme and they named their company as Perfect Day. So now we are going to see what technology do they use. What is interesting that they are using to have an animal free dairy. So once they have the proteins identified, that it is casein and whey, they, if you mix this in water and the nutrients, then you get the milk. So to be put it in a simplest of terms, you can say normal cow milk technology is produced by impregnating cows. Once the youngling is born, the youngling gets taken away from the mother, while the mother gets connected to several machines, starting the process of milking over a span that about lasts roughly about 1 year. The milk they produce isn't effective in converting the nutrients they eat. That is, when they estimated it, they found the amount of fodder that is required, the feed that is required for the cow to eat, only 3% of it is effective and produces milk. So then the cows get impregnated again, and the process is repeated. So if you use a genetically modified yeast that can make dairy protein fermentation, it will be easier and simpler method. In case of genetically engineered yeast, yeast is the host cell, and the plasmids are edited to include the cow gene sequence that produces proteins. Once they create the transformed yeast cell, then they can make proteins. There are two main proteins that the cow produce, which are vital, which is casein and whey. What bioengineers have done is they try to make it at the best, most sustainable type of yeast that can create the milk proteins in order to make the milk products at the fastest conversion rate possible. If we can make dairy that tastes good, and it's the same as the traditional dairy at a comparable price point, no one has to compromise, we would now have the dairy we love at the same availability, the same convenience as before, just as much better use of resources. So if you see the diagrams here, this is from their own website. So genetically modified yeast they have used, and in the process of genetic engineering, the plasmid is extracted, the gene edited, the plasmid is extracted, the gene is edited. Okay, the section of the DNA inside the plasmid is cut off. Then the DNA sequence that the cow used to produce milk proteins is inserted into this particular slot or in the cell. The plasmid is then introduced into the yeast, and the yeast divides and producing. It transfers the genetic information to the chromosomes of the yeast cells. The yeast cells then begin dividing and producing casein and whey protein. So it is so very easy. Yeast is the host cell. Plasmids are edited to include the cow gene sequence that produces the proteins, and the transformed yeast cell can then make proteins when it is inserted. Okay, so this was a simple technique which is used. The below diagram shows that they have extracted the genes, they have modified it, and put it back again into the yeast cell, and then they have grown the yeast cells together. Okay, so next, further, genetically modifying yeast can make dairy products. They came to know the fermentation was easier, a simpler method. The protocol was already produced, but they have to produce this in large amounts. How are the large amounts of these proteins produced? The modified yeast plasmid cells that now have the ability to make cow proteins are grown in large bioreactor tanks. In the bioreactor tanks or fermentation vessels, the yeast cells are given the necessary nutrients, optimal temperature, pH, salinity, and they need that. Then they need to produce the dairy proteins or casein and whey. Once the yeast cells are fermented to grow the proteins, they go through a purification process to separate the pure proteins from the GM yeast cells. The yeast cells are removed off, and only the casein and the whey is taken. Then it is packaged and ready to be shipped to produce the byproducts like ice cream and cheese. So the fermentation tanks that they are using, it's a closed system, isolated from the outside world, absolutely sterile, purest, safest milk protein that is produced. And this milk protein is proved and approved by the FDA. No traces of lactose, no traces of cholesterol, hormones, antibiotics, or anything else that typically comes along for the ride in the cow's milk. So a better way. The result is a protein like no other in the world. So it is identical to milk protein, packing more nutrition per gram than anything else. And it is perfectly done except for the traditional dairy processes that are used. So it is 100% animal free without with uncompromised taste. So next, with what distinguishes the perfect day? This is a technique that they used, but now they are going to use some recent techniques also, or developed techniques also. So what distinguishes the perfect day's milk from other milk alternatives in the store, as is casein and whey proteins that are molecularly identical to the same proteins in milk that comes from cows. They create those dairy problems or sans cow by inserting a modified version of cow DNA into the yeast microflora, then instructing it to produce whey and casein when it ferments from sugar with the sugar. So this biotech company has used, it's a startup, it uses a biotech 3D printer to place the DNA into specific location in the yeast. That is the new technique that they have developed. 3D bioprinting, if you define it, is a method of printing structures with the use of viable cells, biological molecules, and biomaterials together. In simple words, 3D bioprinting is a deposition of biological material in layer by layer fashion to create 3D structures. And here in this particular process, we use bioinks, which are biological materials used in the manufacture of engineered live tissues by the process of 3D bioprinting. The term bioink doesn't only indicate the cells used in the manufacturing process, but it also has carrier molecules that has to give a support to the growing cells. So commonly used carrier molecules, which are called as bioinks, are during the printing process are biopolymer gels that act as 3D molecular scaffold or layers so that the cells can attach, grow, and increase. The polymers used in bioink are essential as they retain water, which provides mechanical stability to the engineered tissue. The process includes 3D bioprinting cow DNA in three-dimensional bioprinting arrangement of cells, molecules, and growth factors within a confined 3D structure based on an alginate hydrogel bioink using a computer-aided design. So this is a simple definition that they have used. So they make it, they place that particular location, particular part of the DNA into a particular location by 3D bioprinter. So 3D bioprinting print cow gene protein sequence so that they genetically engineer the plasmids of the yeast cell to contain the genes. Then they take out a bit of the plasmid's DNA and replace it in the cow's gene. And then the yeast is put into the bioreactor tanks where it is fed with the nutrients, and the process of fermentation continues. The microflora is filtered, you get the pure dairy products and the pure proteins. The process is of course expensive, and the only way to make it affordable is to scale up the project. So what have they done again? So started almost 2 years ago, the Perfect Day Foods raises about $360 million to make animal-free dairy for creating dairy products without cows by genetically modifying the yeast cells to produce casein and whey, which are the main proteins in the milk. That is the today's report. It has raised nearly more than $360 million. The company uses a special 3D printer to insert parts of the cow's modified DNA into specific location in the yeast, which has been dubbed as buttercup. For emerging technologies, the hype cycle always represents this way. It represents the ups and downs that most new technologies undergo as they move from when they start with an idea, they say that, oh, such a super cool idea, this is the first stage. Then what it's good for, that is the second stage. They find out the function of it, the use of it, the requirement of it, and here then how you can actually make money, that is the third stage. So it normally goes this way. So less greenhouse gas emission, less energy consumption, less land usage, less water consumption makes the product better for the planet. That is the website, a very interesting one. Along with this, now recently they have upgraded all their systems, and they are using something, this is called as Internet of Things, IoT sensors, to improve the process. It's also called as Web of Things. The wide construct of this biotech or biotechnology company is it uses a variety or various procedures for modifying living organisms. They use fashionable usage of gene splicing, cells, tissues technologies, and they have all updated technologies. But the researchers are looking for biotechnology management for over a century. How can you manage all these things? How can you put the data together and underline all the difficulties that have come up? So luckily, IoT has a solution to help the employment methodology at every stage or at the biotechnology management. So technologies aren't perfect, that's what they say. There are errors there when you do it. And using yeast to create dairy proteins is, of course, revolutionary in the face of dairy, but the process is expensive and prone to errors. So they use IoT sensors to monitor breweries, possibly cutting down the production cost and making the yeast protein producing technology more advanced. IoT technology will impact how to ferment the yeast. One of the hardest parts in the fermentation process is monitoring and production of proteins in the bioreactor tank. Everything has to be monitored during the fermentation process, that yeast is grown to produce. So yeast requires, this particular process requires monitoring and understanding how the fermentation goes on. Having an IoT here at this particular program, you will find that it is interconnected. This particular sensors are all interconnected to the fermentation vessel lab, and they provide many benefits, including few maintenance costs, then less maintenance, less understanding of how the yeast grow better, because everything is seen on a digital scale, on a digital screen. So the cost, of course, comes down, and how the yeast grows better, and how to monitor the temperature, behavior of the cells, more accurate analysis of conditions in the brewery, then few emergencies, the emergencies become less, and you can optimize the inventory of resources, able to monitor the protein conversion rate, the nutrient intake, the microorganisms growing, the other factors that affect the fermentation, from the yeast during the fermentation. So all this can be monitored on a screen which is connected to the sensors. In most technologic term, you can say interconnected system of both mechanical and digital computing devices, which uses sensors in machines, animals, and even people to create unprecedented amount of detailed data on important systems in order to make them better. I just have, for an understanding, I just have put 3 interesting diagrams or pictures here, which shows a cell comparison of IoT device, microchip inside a human body, with nanoparticle. That research suggests that with right encouragement and understanding, IoT crowd could change their hardware from silicon to bacteria. So they have compared how the bacteria, when it is connected to a system, digital system, how does it work. So in few years, hopefully, you will find most of the chips which are inserted will carry on this information, our information together. So, okay, this is after finishing with the perfect day, I just wanted to put few examples of novel ways of fermenting yeast with a 3D bioprinter to create ethanol. Ethanol is everyone knows, renewable chemical compound made from corn and other plant materials. And it can be found in alcoholic beverages, motor fuel, food additives, beauty and clinical or cleaning products. The 3D bioprinter can be used by freeze dried live biocatalytic yeast cells known as Saccharomyces. So what they do is they have this printer with the Saccharomyces as a bioink along with hydrogen. So these are the complex structures that they produce, and then they use it to ferment, to convert glucose to ethanol and CO2. The process is enabled by a 3D printer, hydrogel lattice. It is the diagram makes it very clear how they use it and how they are processing it. And it is developed on hydrogel material themselves with 70% of water and 30% of polymer infused yeast. So they have a polymer along with the yeast in it, and they infuse it together and make it into a scaffold or a layer. A unique 3D printer is used to extrude this particular, and it is of the consistency of a peanut butter, you can say, which is layered. And then to make it into a cube, it is ultraviolet light is focused on it to make it solid, and give it a shape. Now you have the cubes of 1 centimeter of each in diameter. So it is concentrated form of cells together in a form of a cube. So these 3D printed cubes are then placed into the solution of glucose in order to ferment, just as they would do in brewery settings. So the unique bioink substance allowed for self-supporting as a self-supporting structure has high resolution and high catalytic activity. The bioink material is porous. Um, it develops into a porous scaffolds, enable the conversion of glucose very easily and by the microbes. Also, 3D bioprinting allows for control over the cell density, placement, and structure in the living material. These properties can increase the production rates of ethanol and carbon dioxide. This approach promises to make ethanol production faster, cheaper, cleaner, and more efficient. Researchers from the Lawrence Livermore National Laboratory, LLNL, have used 3D bioprinting even to produce live yeast cells capable of creating a similar substance to beer. This was their first demonstration using immobilized cells. And there is one more that I'm showing you right on the right hand side, which is a phenomenal fermentation, print a drink process, which injects microliters of edible liquid into what it claims the world's first 3D bioprinting process for drinks and other liquid foods. So they have won an award. There is one more company in an Austrian group of company which has been awarded for the use of robotic arm to 3D bioprinter drinks, to use in 3D bioprinter drinks. So print a drink process is perhaps the future that you see, and you can just place it in the solution of glucose and you get the fermentation done. Phenomenal fermentation carried out. I would like to touch upon another company which is of very interest and very fascinating technology that they use. This is company is Biomason. So anyone who has ever heard the story of 3 pigs building their house in their childhood knows that bricks and blocks make resilient structures. So if you know that cement is the second most consumed substance on earth after water, and that manufacturing processes for the estimated about 1.23 trillion bricks made each year results in about 800 million tons of carbon emissions. So this North Carolina based startup Biomason Inc. Aims to reduce carbon emissions by using nature to grow cement. They call it as biocement, more specifically. So Biomason Inc. operates as a biotechnology startup manufacturing company in building and construction industry. It manufactures biologically produced cement building material, and by employing natural microorganisms, non-pathogenic and chemical processes. The company was founded in 2012 and is based in Research Triangle Park, North Carolina. So Biomason tiles are available. As you can see, I have displayed all the pictures that you find on their web page or website, and you can find them in different shapes and sizes and colors, natural and medium gray color, 3 finishes, smooth, horned and polished, decorative inserts, then you find surface aggregate textures, then you find vertical facing assemblies, and installation on a support wall with adhesive systems, and you find it in horizontal condition on a custom base, both exterior and interior use. So you find all of these on the website, and all these are biologically produced. What makes it different? So concrete is the most consumed substance in the world, second only to water. They always compare their company's product with the most developed cement that you find here in US, that is Portland cement. So Portland cement is a core ingredient in the traditional concrete that is used. The cement industry accounts for approximately 8% of global carbon dioxide emission each year, and you require more than 4 million tons of cement that is produced. So building our world's living spaces and infrastructure destroys our environment and kills our chance of the future. That's what they say. So radical change must happen, and it should happen on a massive scale. So Biomason uses natural microorganisms to grow biocement, similar to the way as coral reefs are formed. Sand is placed, you can see the diagram on the left hand side. Sand is placed into a form or molds to which bacteria are added. The bacteria is then fed with calcium and other required nutrients, which allows it to make calcium carbonate between the grains of the sand, stitching all the grains together, assembling them together in required shape. So nutrients and minerals required in the process are obtained locally, naturally, renewable sources. The microorganisms are obtained naturally, they are non-pathogenic, and this process is all completed in ambient temperature, no rise in temperature at all, without the need of high temperature or the traditional manufacturing companies require. A large contributor to carbon dioxide emitted by the processes. No curing is required, just biologically controlled structural cement. To define, these thin masonry biocement tiles are a biologically derived carbon neutral alternative to traditional cement, and they use a proprietary technology that is non-pathogenic microorganism to grow biocement, and eliminating for firing, no rise in temperature, and ultimately reducing carbon dioxide emission. They have labeled recently their tiles as BioLith, BIOLITH, which are made up of 85% of pre-consumer recycled natural aggregates and 15% of calcium carbonate, which is biologically grown limestone. So they grow it on a vibratory compaction system, semi-dry mixed together. They have full strength, and the full strength of the tile comes out within less than 3 days. And very important, they produce it at ambient temperature at 0 carbon emission, and it has full strength as the traditional cement has. An intelligent way, an excellent way, you can say, example of fusing technology and biology together, a new era of construction, right? So coming to the slide where they show all the advantages: grown in ambient temperature using 0 carbon emission process, CO2 neutral biological carbonate cement, 3 times stronger than the concrete block, 100% recyclable or reusable, and every kilogram of biocement used instead of Portland cement, they eliminate 1 kg of CO2. On the right hand side top, you can see the founder of the Biomason, a woman, Ginger Dossier, and who was an architect student of Auburn University when she studied biomimicry and began to consider how natural processes could be applied in architecture. Looking at nature's durable structures like shells or the corals, she saw potential in durable structure cements that provide a sustainable alternative to existing man-made materials. After years of trials, she succeeded in growing a usable brick, which led to the establishment of the company Biomissing in 2012. On the left-hand side, the picture, she has got a government contract. The company has got a government contract and supported by United States Air Force Department of Defense, which developed a prototype engineered living marine cement technology. This deployable biocement is seeded with their consortium, which is their patented self-sustaining natural marine organisms. And this is sourced by nutrients that is found in seawater and propagative, that is, it produces calcium carbonate precipitation. It results in sustained structural integrity, self-healing abilities, and promotion of anchoring to marine sediment floor. You can see the two pictures there, one the dusty road after flight takes off, and the other on the right hand side, which is treated and which doesn't have any dust raised. So after 3 years about laboratory development and successful proof of concept trials has led them large-scale field evaluation using native seawater in natural complex marine environments. So this is the marine that they have done, and they have got this contract. Very interesting, very encouraging also. So coming back, we have two companies. There are a few more companies which do very interesting tasks or very interesting technologies using biology and technology together. So it will be very difficult to remember 2020 as anything other than the year COVID-19 drew the world to a socially distant standstill. But while thousands of life scientists pivoted to trying to understand how the novel coronavirus wreaks havoc on the human body, there were others who transformed their labs into pop-up testing facilities, and there were still more others which used this particular technique, and the field of CRISPR gene editing came up like this. It not only persisted, but it triumphed. And to tie it up all in the bow, you can say that the potential evolution of genome enhancement technologies may likely result in a new era in industrial biotechnology that results in a sustainable bio-based economy. The biorevolution has a potential to help address most of the critical global challenges from climate change to pandemics, chronic diseases, worldwide food security. Yes, biorevolution with its convergence of science and technology has created an explosion of research projects in science and business. If each year the amount of intellectual property related to biorevolution is increasing. This can be seen, for example, by the number of patents that are put up in CRISPR on plant biotech mostly. So in short, the revolution is gaining momentum and holds a promise for industries in all sectors. Thank you so much patiently hearing my talk.